Welcome to part 1 of 3, looking at chapter 7 on cellular respiration. Cellular respiration is the process by which living things obtain energy from organic molecules. So very basically, this is how we're going to create a bunch of ATP from the food that we eat, specifically carbohydrates, the proteins, and the lipids. Those carbohydrates, proteins, and lipids, they all have energy stored up in their chemical bonds, and that's our potential energy. So we're going to take all this potential energy and convert it into energy that the cell can actually use to run other metabolic reactions. So we're kind of going to be burning calories or burning up our food, but it's not really burning like a fire, because generally bonfires, they have a lack of control, they produce a lot of heat, so a lot of unusable energy. We want to convert as much of that energy from the food as we can into ATP. So this cellular respiration process has very extreme control to it. So we're going to be looking at cellular respiration, so how we get ATP created from our food when oxygen is present in the environment. That's going to take up the first two sections of this chapter. It's a pretty long process. The third part of this chapter, we're going to be looking at anaerobic respiration and then also a process called fermentation. And these are things that happen when oxygen is absent. And we'll talk about when we have oxygen absent from the environment. So cellular respiration, again, it's a process by which living things obtain energy, and we're going to get that energy and put it into the form of ATP. And we're getting this energy from all the potential energy stored in organic molecules, specifically from the food we eat. And then in addition, we're also going to create some waste products that I'll show you. Again, the primary aim of this whole process is to make lots and lots of ATP. Which the cell can then use to do pretty much everything else within the cell. And we're going to be looking at aerobic respiration. Aerobic means with oxygen, so this process is going to use up oxygen. So pay attention to see when the oxygen actually shows up in the process, so you know how to answer that question about the oxygen. Okay, so we're going to be looking at cellular respiration, starting off with the molecule of glucose. Glucose is a monosaccharide, so it is a carbohydrate and just a really simple carbohydrate. It's a hexosugar, so our glucose is going to be the C6H12O6 molecule that we see up on the screen. So we're going to take that glucose, we're going to add in 6 molecules of oxygen, because remember it's aerobic with oxygen process. We're going to go through a bunch of steps, so a whole bunch of different chemical reactions, and then out of this we're going to get a waste product of carbon dioxide, which we end up breathing out. We also get water that's produced, so the H2O. And then the thing that we're really interested in is the ATP. And we're going to get up to 38 ATP for every 1 molecule of glucose that we started off with. So we really want to make all of these ATPs through this process. So I mentioned a couple of times that cellular respiration is a very, very long, complicated process. We break it down into 4 metabolic pathways, and we're going to go through these metabolic pathways. So we start off with a glycolysis. Number 2, we get to the breakdown of pyruvate to an acetyl group. The third part of cellular respiration is the citric acid cycle, and then we're going to have oxidative phosphorylation. On the previous slide, we went through kind of the overall equation of cellular respiration. I talked about there are 4 steps that we're going to be looking at, and these 4 steps make up this whole process of cellular respiration. This slide right here, this is going to be the most important slide of this chapter. It basically summarizes the majority of the information that we're going to be covering. So I'm going to spend a little bit of time going over this slide up here. So the first thing I want to point out, in the background, you can see there's kind of an orangish brown rectangular structure. That is the mitochondria. And if you remember in chapter 4, we talked about mitochondria, and they're the powerhouse of the cell. They're responsible for doing cellular respiration. So this makes sense. Now we have our mitochondria and the cellular respiration process up here. In addition, if you see, there are 4 light blue boxes. Those are going to be the 4 different pathways we're going to be looking at. So up in the left-hand corner, we have glycolysis. We have the breakdown of pyruvate. The citric acid cycle is number 3, and then number 4 is our oxidative phosphorylation. Okay, so this image, why it's so important is it's going to tell you what goes into each of the 4 steps. You're going to see the inputs for your 4 different steps. You're also going to see the outputs, so what comes out of your 4 steps. And then the last thing is it also tells you where each of these different 4 steps actually occurs within the cell. And I'll be going through these inputs, outputs, and location as we go through the 4 steps. But just to give you an example, if we look at glycolysis up in the left-hand corner, you see glycolysis, the first thing that occurs. So our inputs, we're going to have glucose, which is that 6-carbon hexose sugar that we talked about in the previous slide. So this glucose molecule is going to go through a bunch of different chemical reactions, and out of glycolysis, if you notice there is an arrow coming out the bottom of the blue box, and it splits off into 3 directions. So if we look at the first black line, the first arrow going off to the right, if you follow that, there's a 2-NADH coming out. So that means that glycolysis is going to produce 2 NADH molecules. And if you remember, NADH, these are one of those energy intermediates that I talked about in chapter 6. If we look at the black arrow coming out of glycolysis again, you have another arrow that splits off that says 2 pyruvate molecules. And just to point out, those pyruvate molecules go into step 2, the breakdown of pyruvate. And then in addition, if you follow the arrow going straight down, we also get 2 ATPs that are created. Okay, so this is kind of how you're going to use this chart or diagram up here. And location, glycolysis occurs in the cytosol. The other 3 steps, they're found in that mitochondria that's in the background. So again, it's really good. You have your inputs, your outputs, and where this process actually occurs in the cell. The very last thing, if you look back to those 2 ATPs coming out of glycolysis, if you look across the bottom of the diagram, there's 2 more ATPs from the citric acid cycle, and then we have 30 to 34 ATPs coming out of the oxidative phosphorylation on the right-hand side. So we can get up to 34 plus 2 plus 2, so that's where we get our 38 ATPs for 1 molecule of glucose. Those ATPs, right below them, it tells you how those ATPs are actually created. So we have that substrate level phosphorylation for glycolysis and the citric acid cycle, and then the 30 to 34 are made using the chemiosmosis process. Just to quickly review the 2 types of ATP synthesis, we have our substrate level phosphorylation, where we're going to be using an enzyme to directly make our ATP. So that's, again, we get 2 ATPs from glycolysis in this method, and then the 2 out of the citric acid cycle using this method. But the majority of the ATP during cellular respiration are made using the second type. And the second type is that chemiosmosis. And I'll just quickly run through this again. We'll see it come up when we get to the 4th step, the oxidative phosphorylation. But here is the chemiosmosis. We're going to have energy that's stored in an electrochemical gradient. So we have to have a membrane set up. On one side of the membrane, we have a bunch of hydrogen ions. So in this example, the hydrogen ions are on the top of the membrane. On the other side of your membrane, you have fewer hydrogen ions. Remember, molecules, they want to go from high concentration to low concentration, because that's just how the universe works. Things want to go from being ordered to being more disordered. So they go towards that entropy. So these hydrogen ions, they're ions, so they can't just passively diffuse across the membrane like other molecules. They have to use a facilitated diffusion, so they have to go through a protein channel to get across the membrane. So that protein channel is going to be the ATP synthase, and as the hydrogen ions go from high to low through the ATP synthase, it's going to synthesize ATP. Okay, so remember this picture here, this diagram, this is going to summarize what we're going to be talking about in pretty much this whole chapter. So we're going to start off with the glycolysis part, so the first step. And remember, really focus on the input, so what's going in to that step, the output, so what's coming out of it, and then where it's actually happening inside of our cell. Glycolysis can occur with or without oxygen. Remember, we're going to look at it with oxygen at first, and then we'll come back to this process when we get to the without oxygen and the fermentation part. Glycolysis, like I mentioned, occurs in the cytoplasm or the cytosol. So remember, that's the location. And this process, glycolysis, it has 10 steps to it. And these 10 steps are broken down into 3 phases. So we're going to have an energy investment, so we have to put a little bit of energy in to get the molecules ready and started. There's a cleavage phase, and then energy liberation, where we're going to get out our 2 ATPs and our 2 NADHs and our 2 pyruvates. These 10 steps in glycolysis, they're really, really important because these 10 steps are pretty much identical in all living things, whether if you're looking at bacteria, plants, animals, fungi, protists. These steps are very, very common in all of these different types of cells. So this process is pretty universal for all living things, no matter where they live or what they are. The first phase is going to be our energy investment phase. We're going to invest 2 ATPs that we're going to hydrolyze. We'll add water to them. That causes the ATP to let go of a phosphate and become ADP. So those 2 ATPs that are broken down, they also release energy, and we use that energy to convert our glucose molecule into a molecule of fructose 1,6-biphosphate. So I'm going to show you this process. This is the first 3 steps out of our 10-step glycolysis process. So we're going to start off with a molecule of glucose right here, which has 6 carbons to it. And then we have our first ATP molecule. This ATP molecule is hydrolyzed by an enzyme called hexokinase, and we get a glucose molecule with a phosphate group on the 6 carbons. We get a glucose 6-phosphate. So now we have a yellow phosphate attached onto our glucose. So this is our first step. That glucose 6-phosphate then goes through a second step where it's basically rearranged into a fructose molecule, which is also a 6-carbon sugar. So we have a fructose 6-phosphate. And there's an enzyme that specifically does this step, so the phosphoglucoisomerase. It just rearranges some bonds to create a fructose 6-phosphate. We then have our second ATP coming in during the third step, and this ATP gets hydrolyzed by the enzyme phosphofructokinase, and we end up with our fructose 1,6-biphosphate. So very basically, we have our 2 ATPs being invested during step 1 and 3, and our glucose was arranged into a fructose. So we end up with a fructose molecule that has 2 phosphates attached. One phosphate is on the first carbon, the other phosphate is on carbon number 6. This fructose 1,6-biphosphate that we just created during this energy investment, it then goes through cleavage. So fructose, like I mentioned, it's a hexo sugar. It has 6 carbons. So this 6-carbon molecule is going to be broken down into 2 3-carbon molecules. And those 3-carbon molecules are called glyceraldehyde 3-phosphate. So I'm going to show you this process. Okay. So we have our energy investment, the first 3 steps. We then take our fructose 1,6-biphosphate, that is kind of on the right-hand side, and it gets split in half into 2 glyceraldehyde 3-phosphates. And it can be converted back and forth between a dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So that's why we have kind of the split on the right-hand side. But basically, split our 6-carbon molecule into 2 3-carbon molecules called glyceraldehyde 3-phosphate. And here it is showing that those molecules can all be converted into the glyceraldehyde 3-phosphate molecule. So that's the one that we're interested in during this cleavage phase. Those glyceraldehyde 3-phosphate molecules that we just created during the cleavage stage, we're then going to break them down into the pyruvates. Remember, pyruvate is one of our outputs. As we're breaking down those molecules into pyruvate, we also get 2 NADH molecules, so 2 energy intermediates, and we also get 4 ATPs. So remember, we get 2 ATPs out of this glycolysis. So remember, energy investment, we invested 2, so we have a -2 plus 4, -2 plus 4 equals 2, so we get a net yield of 2 ATPs from this process. So we're going to take our glyceraldehyde 3-phosphates, they go into another step. This is where we get our 2 NADHs that are created. So you can see the NADH being created during this step. And we end up with a 1,3 bisphosphate glycosyrate right here. And you have 2 of those. Those molecules are used to create ATPs. So we get NADHs created, then we get 2 ATPs. The molecule's converted into another form. We convert it further, and eventually we get our other 2 ATPs. And we end up with pyruvate at the very end. So I know it looks pretty complicated. Again, remember, really focus on the inputs, the outputs, and where this is occurring in the cell. And basically this process is just showing you that each of these 10 steps, they have their own enzyme to them, because remember, enzymes are very specific to what they do. So each enzyme does one little part of this whole glycolysis pathway. So this glycolysis pathway, like I mentioned at the very beginning of this chapter, cellular respiration is very, very controlled. So here's an example of control during glycolysis. If we have a sufficient amount of ATP already in the cell waiting to be used, that ATP can actually go back to the very beginning of glycolysis, and it can turn the whole glycolysis pathway off for a little bit. So here's a real-life example of that feedback inhibition or negative feedback that we talked about in chapter 6. So if we have a lot of ATP, we have a lot of the end product building up. That end product will go back to the very beginning of the pathway, or close to the beginning of the pathway, and it will turn one of those enzymes off. So specifically in the 3rd step, we have our ATP. It's binding to an allosteric site on the enzyme responsible for the 3rd step of glycolysis. And remember, if you have something, an inhibitor bind to the allosteric site, it's going to change the active site on the enzyme, and it's going to shut that pathway down, shut that enzyme down. So we have an actual example of one type of this feedback inhibition that we talked about earlier. Okay, so that was glycolysis, and again, remember, we put in glucose, it goes through energy investment, cleavage, the energy liberation part, and out of it we get 2 NADHs, 2 energy intermediates that we're going to use later, 2 pyruvates, and 2 ATPs using substrate level phosphorylation.